Yep, all right. Okay, for those of you who came, I hope you'll find this fruitful. Dr. Bolter mentioned that this is not an English class, and I agree, but, that's a huge but, one of the things I feel that you should be getting out of your undergraduate experience is the ability to communicate well in writing. And so a week from Tuesday or a week from Wednesday, depending on which lab you're in, you're going to have your next write-up due. And what I'm going to ask you to do for this is to essentially have a draft of what would be like an article in a journal. And please, full sentences. I saw some of the Bolter labs and they were just bullet points in places. No, full sentences, and there's this time-honored idea of a paragraph, one idea per, and you should also adhere to that. And so that's the, and I can also recommend, who owns a copy of Strunk and White? Nobody? All right, I'll try to find out. There's a link to White. Strunk's pithy commentary isn't in there, but it's a cheap little paperback. It's funny, and it really shows you how to write well. I had to learn how to do this in graduate school. It was very painful, so I would hope you'd learn it now. Okay, so what I'm going to ask you to do again, by way of your assignment, is to write a lab report that is like a draft of a journal article. And so you should have these various parts of it. And I'm going to go in detail about these various parts. You should have, besides a cover sheet, an abstract, an introduction, a method section, a results section, and a discussion section. And usually not, well, more and more anymore, but often not, you won't have an addendum, but you will have an addendum. And what it's going to be in your addendum actually is we're going to take pictures of the tissue that you sectioned. You're going to stain them and take pictures of them regardless of what you got. And so what I'm going to ask you to do is put a picture of your tissue, and also we will attempt to find sections that are illuminating if your mystery bird was a male or a female. And so I'll ask you for some evidence about whether or not it was a male or a female. So you'll also have an addendum. And, ooh, I can see, need to fix this slide right here because there's no reference section here listed, but there ought to be. All right, so you'll also have a reference section. Okay, so an abstract. Let's talk a little bit about what one would put in an abstract. Quite honestly, I write my abstracts last after I've written the whole paper, because your abstract is basically a verbal map of the paper to get the reader through it. So I often don't know where I've been till I've been there, so I usually write the abstract last, okay? But it doesn't matter. Some people will write the abstract first and use that as an outline to develop the rest of the paper. That's fine too. It should be a thumbnail sketch of the whole study. So what's the big question or questions that you're trying to answer here in this study? You should relay that to the reader. You should briefly, very briefly, you only get about a page for these things, maybe a page and a half if you're getting long-winded. Say what kind of methods you used. Very briefly, the results. I have a strong preference of don't put numbers, like restate all the means, and I've seen that, but it's hard to read and doesn't make much sense as presented like that. So a verbal description of how the results came out, the big picture of the results, not every little hiccup in detail, and then a little tiny bit of the discussion. What do you think those results mean with regard to your question? So it's basically a map of the paper. So the reader can read that and say, okay, now I know the story that I'm going to get into here. All right? Any questions about this? So again, page and a half at the outside for an abstract, okay? And when I rarely do say page numbers, I'm always talking double-spaced. Please don't kill your TAs and strain their eyes. So double-spaced is courteous. Okay. So in the introduction, what you got to do in the introduction is you got to go from the big picture and narrow it down to what you're actually going to do in that study, what we actually did. Okay? So you need to define things, and if you wonder if you need a reference for something, if you turn to your major, who's hopefully not in this class, and you say, tell me about the organizational effects of steroid hormones, and they go, huh? You need a reference. Okay? So what we're going to be talking about, what we're doing in our study is looking at the organizational actions of steroid hormones, and it occurred to me that in the reader I hadn't put any references in the reader that explicitly dealt with that question, but you would need to reference something. So on the course website now, there's a page by Dr. Arnold, who's a professor here, a paper, and I don't ask you to get into that whole paper, but the first 5 pages or so, he does a really nice job of talking about the organizational actions of steroid hormones. It's actually a 50th anniversary paper of when the original hypothesis came out, and I guess I can say I've read the original paper, and it's tough going. He does a lot better with the ideas than the original paper does. So it's a very nice summary, okay? Okay, so you need to talk about, okay, are we examining the organizational effects of steroid hormones? Are we examining in our study and manipulating the activational effects of steroid hormones, or both? So what are we doing here? What were we doing in our study? Organizational, activational? Organizational. They got manipulated way early when they were just babies, right? And then we looked at them when they were grown-ups. Okay? All right, other things you'll need to do. By the way, what I'm showing you here is essentially the grading key. I've taken the points off it, but this is the grading key in essence. I may tweak it a little, but this is it. All right, so you need to define the song system for the reader. You know, what is it? Okay, what's it do? And where could we find these song nuclei? Okay, so let's actually talk about that because I'm not sure you're clear. So the song nuclei, which I am going to show you in a couple of slides, actually right there. Song nuclei. So we measured which ones? HVC, RA, area X. Okay, so what subdivision of the brain may they be found in? Telencephalon. So all telencephalic. Okay. All right. So let's go back to that slide. All right. So where are they? What do they do? Okay, and I touched upon that in my first lecture. By the way, I don't like people referencing my lectures, like Grisham personal communication. No, that's not so good. All right, everything you reference ought to be something that I could download from a library or even go and physically touch the journal in the library. Okay, you don't get to use my jabbering as your reference. Okay, I hope I'm giving you some background here, but you got to go find something in writing, okay? You have something nice in writing about the song system. This is the Notenbaum and Arnold paper, which is in your reader. This was when the sex differences in the song system were first described, okay? And they did a dandy job with it, all right? Okay, we've already covered that slide. Okay, so then in the introduction, again, now you're focusing the reader down to the specifics of what you're going to do exactly. Okay, so you got to outline the project. What are we going to do, and why did we do that? Okay, and so by way of doing that, you need to say, okay, so which hormone did we manipulate when we gave different hormonal treatments? Estradiol, okay. And actually, I'm going to skip to my next slide and talk a little bit about that. And what I want to emphasize is don't just say estrogen, because everything here in this gray triangle, all of those hormones can be referred to as estrogen. So you need to specify it's estradiol is the one that we manipulated. Okay? Going back then, okay, so, and then you probably want to tell the reader, since most people probably don't realize, what's the big masculinizing hormone, by the way? Estradiol, okay. So, and what makes you think that other than stuff I've told you? You have no idea. I trust that you have not yet read the readings in the reader. Okay, so read the readings in the reader, and there will be a paper in there by this Grisham character, Grisham and Arnold, that tested all the different hormones in a nose-to-nose comparison. Okay, so why that one? Well, it worked the best. Okay, and also there's another paper by Grisham et al., a whole bunch of us, who that's essentially the paper that you are replicating, and that's also in your reader. Okay. All right, so why that one? Okay, tell the reader about it and reference it. Just don't say, yeah, we're going to use estradiol because it works well. Well, why? Who said so? Okay, give us a reference, okay, or references. Okay, you want to include predictions and why those predictions are made. You may not always be able to include predictions, but you can try at least. I think you should try. Okay, so I'm going to skip that slide since I've already dealt with it, and let's talk about the main questions that we were trying to answer in our study. Okay, and the big thrust, of course, is going to be to gauge the sensitivity of the female song system to estradiol. Okay, if the reason that I did that study originally was if the female song or the zebrafinch song system is explicitly sensitive to estradiol, I could have saved the hypothesis that gonadal steroids are exclusively responsible for sexually differentiating the system. And I wasn't able to save the hypothesis, and I finally ran away from it myself. It's, in general, I think it has some validity, and for lots of animals, it has a lot of validity, and I'll talk about this hopefully in my next lecture, but it's not the whole story. It needs to be refined. Okay, we also wanted to see if we could replicate sex differences, so you probably want to, at that point, tell the reader that there are some sex differences in this system and use a reference. Okay. We want to see if masculinization is an all or none event once the threshold was reached, or if it shows a graded response. If you want to make a prediction on that based on something you've read, that'd be great. To determine if the parts of the song system show differential sensitivity to estradiol. So we're measuring 3 different parts. Are they going to all show the same sensitivity, show the same degree of masculinization, or is it going to be differential? And you could predict that possibly also. And so you're actually, when in your study, extending my study. And in your study, you have males. I didn't have any males in my study. But we're going to see, or you're going to see, if your largest dose is of estradiol is going to masculinize the songs, fully masculinize the song system of females up to male levels. You'll be able to make that comparison, okay? And if you want to make a prediction based on literature that you have at your disposal, you should do that, okay? All right, any questions about this? All right, so at that point, we've pretty much done the intro. We've gone from the big question of the organizational effects of steroid hormones. We're now narrowed it down to the song system, defined the song system, made the predictions, explained to the reader why we're doing what we're doing. Okay? Okay. This is an option. Mostly what I've talked about so far would not be optional for a really good lab report. Some writers, and me included, at the end of the introduction will give like a sentence or two that explains to the reader how it came out. Some writers hate that, all right, and say you shouldn't do that. But I kind of like it because it gets the reader on track, like, okay, once we hit all this method section, and I don't know about you all, but when I was undergraduate, I had a hell of a time reading through those method sections, okay? So it kind of helps the reader skip from the intro. If the method section gets them lost, they can get into the results and discussion and make some sense out of the darn thing, okay? So I kind of like a couple sentences there. That's a complete option. I'm not going to put it on the grading key, but to me it's a good way to write so you keep the person on the track that you want them on. All right, so that's it for the intro section. Okay, let's talk about the method section. Okay, you need to describe the birds from which the data was taken. And that Grisham et al. Dose response study, your birds are those birds except for the males, but the males, other than being male, had the same characteristics in life and everything. They were the guys living in the next cage. You can use the information that's in mine. Put it in your own words, put it in your own words, put it on your own words. Don't just copy over mine and paste it in yours, okay? Some things you need to emphasize. So I am actually sure that your N sizes don't always match mine, okay? You all know what I mean by an N size, right? Okay, so the number of birds, okay? And the other thing that you really need to mention is you have males in your study, which I didn't, okay? So you need to mention that you got males, all right? This should be a shorty paragraph, okay? Not much. Okay, the next subsection of the method would be apparatus, and I'm going to beg you not to put in the detail that I had. All right, all you need to do is say that the measures were taken from digitized images taken through a microscope for area X, HVC, and RA, and that we used NIH image. And NIH actually stands for the National Institute of Health, so this is free software that they give away. You could just Google image J and get your very own copy if you wanted to. All right, your tax dollar at work. So you don't need to go into all the painful detail. That ought to do it. Again, sentence or two. All right? Okay. All right, the procedure. Again, you're going to get most of this out of that Grisham et al. Dose response article, but put it in your own words, put it in your own words, and put it in your own words, okay? You can paraphrase me. By the way, let's talk about that a little bit. Students think if they change one word in a sentence, that's a paraphrase. Is that a paraphrase? No, 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 no. All right, so that's not a paraphrase. Even two words may not be a paraphrase. Put it in your own words, okay? One things that you're going to need to do is describe the estradiol treatments and the control treatments well. What were the doses? How were the pellets made? Maybe I can stop and talk a little bit about that. The pellets. What we did was we made pellets that would give us kind of a slow release of estrogen, estradiol, I should say. And to do that, what we traditionally do, as I describe in that article, is you take this elastic glue, which is kind of like, you guys ever see aquarium sealant or something like that? It's very similar, except for this is medical grade. Okay, you can put it in a body because it's been filtered, all right, and it's all cleaned up and niced up. So what we do is we take that glue, we crush up the estradiol, get it really fine, and mix them together, and then you stick it in a syringe and make what we call a rope. So you just take a syringe, I think without a needle, that's how I did it that time, and you squeeze out a line of this stuff. And then you weigh the line of this stuff that you make, and you say, okay, so I know it's so much percent estradiol because I know the weights that went in, and I can cut it up and make it into, say, 50 microgram doses or 5 microgram doses or whatever. Okay, you don't need to go into all that grievous detail that I just gave you, but that's how you do it. Okay, so we weigh the pellets and make sure that we have the appropriate dose. And of course, you know, I'm sorry to say I'm not perfect. I think we tried to get our doses within 10% of the ideal. Okay, it's about as good as you can do. Okay, so you need to say, you know, the description of the pellets. You have to talk a little bit about, especially about the days. What day did they get those doses? What day of, like, was it in the egg after hatching? What? Okay, and you have to also talk about the controls. All right, and controls all got an implant, but they didn't have estradiol in them, okay, including the males. All right, so the male and female controls got a pellet implanted, and the pellet, by the way, they don't seem to mind much. You just stick this thing right under their skin on their chest, they're fine with it. All right. Do y'all ever hear about Norplants? No? Okay. So this is the kind of birth control that they use in the third world, and it's really similar to these pellets in many ways. So what they do is they just put hormone in a slow-release kind of celastic capsule, not a glue this time, and then inject it in women, especially in the upper arm. And so that's all the birth control you have to take for a year. Every year you have to get them hauled out and replaced. But that's what they're essentially a Norplant, zebrafinch size. Okay, so everybody okay with that so far? All right, make sure that the reader understands we're using estradiol. Don't say estrogen, because there are at least 4 compounds that could be estrogen, okay? It's estradiol. It's E2. You can say E2. That's okay, too. All right. As long as you define it somewhere, all right? Okay, you got to define it early on, and then you can use it. Okay, and again, because estrogen would refer to all this stuff, we don't even have E4 on this diagram. But you want to make sure that the reader understands which compound exactly. Okay, so the brains that you have the pictures from were cut at 40 microns. They were frozen section just like you did the other day in lab, and they were stained with thionin. And in lab, you're going to get to do this also. Please don't put in all the details, like every step of alcohol and water that we put our sections in. If you just say stained with thionin, people can look on the web and see what a thionin staining protocol is. You don't need all that kind of detail. I would, however, ask you to tell me what thionin does. Okay, so what's thionin stain? It's rough ER. It's what's known as Nissl substance. And if you go way, way back in the literature before Nissl, this stuff was called tigroid substance, because it looked like tiger stripings in the cell. And I don't know how Nissl managed to get it named after himself, but he did. But anyway, we now know it's rough ER, and it defines the cell bodies. Okay, and what do I mean by that? You can see the cell bodies, and what can't you see? You don't see the axons or the dendrites. Okay, and actually, I used to think once upon a time that you could what we actually were measuring was the cell bodies, but you can't. Turns out you can't see the cell membrane with the light microscope. But the good news is apparently the rough ER goes right up to the membrane. So we're defining the cell bodies. Okay, so I do want you to tell me what a Nissl stain does. All right. Okay, you should describe how the areas were measured. That is to say, you should give the reader the units, okay, to the nearest what, okay? If you don't know, you might take a peek when you get back to the lab, all right? And we did something a little bit different than I did in my study. What we used were summed areas, okay? And I did that for a couple of reasons. So when we just sum the areas across both hemispheres, you don't have to sort out which one came from the left and which one came from the right, okay? And the other thing is learning from bitter experience. There's like 2 math steps you have to go through to make an area turn into a volume. And my students were really great. My previous students, I'm sure you guys wouldn't be like this. My previous students were really great at screwing that step up. And so I thought, we'll just sum the areas, and we'll get the same kind of results, and we won't get any wacko data. Okay, so unlike Grisham et al., you didn't do volumes, you did summed areas, okay, and you need to tell the reader that. Okay, the measures were done blind with regard to sex and treatment, so you need to say that. And you wouldn't usually in a journal article say why that's a good control, but part of what I'm trying to teach you is what good controls are and why we need them. So please put that in. So why would you do it blind? Why would you make it bigger or smaller? Yeah, and so what are we trying to control for? Bias, okay? So if you know, even the most honest person could subconsciously be drawing it as the way they want it to come out, okay? So any study I do, or if I have my students measuring for me, I always make sure they have no clue who's in what group, all right? It's really, really important. So I want you in your lab report to tell me why that's a good control and what it's controlling for. Okay, and this is a little subtle behind-the-scenes kind of thing that I built into this study. So each of you I'm going to call an observer, all right? And you got 5 birds, and I think I even told you up front. One was a control male, one was a control female, and then you got one from each of the different estradiol conditions. So you got a 0, a 5, a 15, and a 50, right? So you should have ended up with 6 birds. Everybody get 6 birds? Hope so. All right, so why did I do it that way? So why not say, just give one person all the males, another person all the females, different person gets the 0, different person gets the 5s, another person gets the 15s, another person gets the 50s.
Somebody has a tendency, certain tendency when they're drawn in the area, then you want them to carry that to both sets of data.
Exactly. So each of you is a wonderful, unique individual, and what that means is you do things in wonderful, unique ways. And what that means is for our data is you're going to wonderfully inflate the variance, okay? And as I watch you, I see, so say here's area, or say this is RA, okay? This is how it would be, this is how it is in reality, okay? So as I look over your heads, I see Steve Stingy, he's drawn it like this, okay? And I see Laura Largess, and she's drawn it like this, okay? All right, so say I gave Laura all the males and I gave Steve all the females. At the end of the day, I get a difference. What can I attribute that difference to? Is it real? You can't tell. This study would then be what? And screwed up is not the word I'm looking for. What would it be? There's actually a real official title for this kinds of problem. Say it loud. Invalid. And what I'm trying to get you to say is what?
Spurious.
Spurious is actually when you get a positive that shouldn't be there, okay? So here we don't know. We have a difference, and we don't know, is it a real difference? Is that in the tissue, or was it how they drew it? Okay? And so we're confused. And another word for confused, we don't use it much in English anymore, is confounded. You've heard this term? Okay, so then we'd be confounded by, we'd have the treatment confounded with the observer. So we don't know at the end of the day, was it real? Was it how their personalities, how they drew it? Can't tell. All right, so what I did instead was to make sure that didn't happen is I gave each of you, in all of your wonderful, unique ways, a bird from every group in the whole study. Okay, so then at the end of the day, if we see any differences among our groups, we can be fairly confident it's not confounded with how people drew it. Okay, does that make sense? All right. This is a technique that's called balancing. So if you have some possible confound like this, what you can do is you can balance it across your groups. And so that's what I did, was I balanced it across the groups so that it didn't differentially act in any one group. Okay? It's called balancing. And for those of you who took Psych 100B, not too many of you, a couple of you, all right, so there's a close relative of this called counterbalancing, okay? And so counterbalancing has the same idea in mind, but it's not quite the same. So counterbalancing you might see in a clinical drug trial. So some of the people start on placebo and shift to the real drug, and some of the people start on real drug and shift to the placebo. So that's That's called counterbalancing. All right. This is just balancing. All right. So I'm balancing this possible confounding effect across the group so it doesn't differentially operate, doesn't confound my experiment. So you need to tell me about that in your lab report. While we're thinking about this, and this is something I'll come back to probably more in my next lecture, is, as I pointed out, you guys you're doing it all kinds of different ways, all right? So what will this do to the variance in our data? Will it make it small or make it large? Large. And in fact, when I do these studies for real, I have one individual trace one dependent variable, because if you put more, even two people on it, you really start blowing up the variance, okay? And as I hope your professors previous to me have told you, variance is bad, right? Okay, what does variance do to your results? Makes them less precise. And is it easier or harder to get significant differences? It's a lot harder, okay? So you want variance to be as small as possible. This is a topic to which we'll return. You want your variance to be as small as possible in order to get significant differences. Well, unfortunately, we don't have a situation where we minimized it, but keep that in mind when we start thinking about our data come Thursday or even Tuesday or Wednesday, okay? So variance was fairly large, certainly relative to the study where I ran it. Okay. All right, design. You need to tell the reader what kind of design was this. So was this between subjects? Was this within subjects? Okay, between. What's that mean, between subjects? Comparing between individuals, and do the individuals, are they in every group or not? No, each individual is in one and only one of the groups, okay? So this is what's called a between subjects design. And of course, when you do looking for sex differences, you're always forced, right? You can't turn them in, take the boys, turn them into girls, girls into boys. Can't do that. All right, so it's a between subjects design. We're going to analyze the data with a one-factor ANOVA, which I will talk more about in my next lecture. You'll get to use it, and then I'll try to give you some insights as to how it works on Thursday. We had one factor. One factor, how many levels? 5, okay. And so those were what? Okay, so control males, control females, 5, 15, and 50 micrograms bestradiol in those other females. Okay, so those are our levels. Now, as I'll talk more about in my next lecture, an ANOVA is actually a really neat design that controls for, especially for getting false positives. But when it comes to the end, if it gives you something, if you get a significant ANOVA, it doesn't tell you which group is different from which other group. Okay? So it's really good at controlling for high rates of false positives, but at the end, if you get a significant one, you know there's a significant difference in there somewhere, at least one, but you don't know where. So what we're going to do is we're going to go back, and if we get a significant ANOVA, and I know at least Tuesdays got one at least. I haven't done all the data, and I didn't do Wednesdays at all. So, but I'm highly confident that your data actually are looking really nice. So you're going to get at least one significant difference, I'm sure, and I suspect a whole lot more. Okay, so you get a significant ANOVA, and that's all I did, by the way, also in your data so far. You want to go in and you want to say, okay, which group is different from which other group? So you have to do some subsequent tests, and you can only do these subsequent tests if you get a significant ANOVA, all right, or analysis of variance. And these are called post hoc tests, okay? And you go in with those if and only if you get a significant ANOVA and find out, those will tell you which group is different from which other group, okay? Post hoc, any Latin scholars here? No? Okay, after the fact, actually. After this is what it means. Okay, and the one that we're going to do is one near and dear to my heart. We're going to do Fisher's LSD test, and that isn't as fun as it sounds. There will be no drugs distributed in this class, but what that actually stands for is Fisher's least significant difference test, okay? And it is, post hoc tests come in a variety of flavors, and from liberal to conservative. And Fisher's LSD test is the most liberal one that's out there. And quite honestly, I like it because when I do these studies, it's usually a year or two of my life. And so if I'm going to pour that kind of time into it, by God, I want to get something out of it, all right, if I possibly can. So I usually use these more liberal ones. It's certainly fine once you get a significant ANOVA, you have license to do that. So I don't have any qualms about it. Oh, between subjects, I'm sorry. Sorry, between subjects. That's an old abbreviation that I should have talked about. Okay, that was the methods. All right, now, any questions about methods before most of what I'm going to talk about results we're going to do on Thursday when you actually have your results in hand. Okay, one thing I'm going to say is some of you took courses where you got a template on how to write a results section. Don't use it. It will, one, screw you up, and two, you're running a strong danger of setting off the plagiarism detector. Okay, don't use templates from other courses. All right, so in the results sections actually are kind of tough to write. You're just going to present the facts. Don't interpret it. Okay, you just want to, you can say things like this group had bigger RAs than that group, but you can't say, you're not supposed to interpret it. That's for the discussion. Okay, you're going to be also directing when you make a statement. You're going to make statements in words. You're going to back the statements up with statistics, which I'll show you how to do either in lab or in lecture on Thursday, and you're going to direct the reader to a graph. Okay, and I need to point out, this is not your data, okay? This is some data from, I piloted out this particular module a couple summers ago, and this is some data from theirs. So there's some aspects of this graph that I really like and some that really stink, even though I made it. So this is a graph which is, and every graph should have a figure caption too. If you want to put the graphs and figure captions in the results section, I don't have any problems with that. Okay. Traditionally, we used to have to put it all at the end of the paper, all right? And that's because when they published papers, they had to take the graphs and send them out to artists and cast them in lead and all this other weird stuff that with computers you never ever have to do anymore. So you don't have to put them at the end. If you want to put them in the text, I'm fine. Do you guys have any strong preference? David and David, where they go? Okay. If you're going to put it at the end, make sure they know that they're at the end, okay? All right, so you're going to refer the reader to a graph, okay? And this graph should have a caption that says, okay, this is the mean sum of RA areas as a function of group. And one of the things I don't like about my graph that I drew, what are some things that aren't so good about this graph? Anybody see it? Yeah. B and B, I'll get around to that. Okay. That's actually something I like about this graph, but I'll get around to it. Pardon? We'll take you first. Units! Got no units on the y-axis, got no units on the x-axis, and I'm sorry about that. I should have put that in. You might get away with putting, how do you guys feel if they put it in the caption? Is that okay? All right. So I would certainly have to put it in the caption, like say what 0, 5, 15, and 50 referred to. Okay, and what kind of units on the y-axis? All right, I need to do that. All right. And you had something else, though? Title wouldn't hurt either, okay? Even a title like Figure 1 would be okay. All right, things I do like about this graph, and something I'm going to ask you to do is put error bars on your points, all right? And I'm going to try and get a how to make error bars in Excel cheat sheet up on the course website. There, you can do it, and it's actually not that hard, but you need to know a couple tricks. All right, so error bars. Why do I like, and I like standard error bars, and you should tell the reader that they are standard errors, okay? Why do I like standard error bars? If you know something about statistics, you can do statistics by eye, all right? I'll come back and talk about that later. The other thing I like about this is all of these groups that are connected by a line are females, and our males are not connected, because you can't have some, having a line suggests some continuity among the groups, okay? And it would be difficult to argue there's some continuity with males, okay? So not having a line there was good. Let's talk about those A's and B's, okay? What I got was the results, which I don't have presented here, but control males were different from every other group, okay? And I think that was P less than 0.01 or something like that. And the 50 and 15 group were different from the 5 and the 0 group, but not different from each other. Let me say that again in slow motion. Okay, so the 50 and 15 group were different from the 5 and 0 group, okay, but they weren't different from each other. So, and that was, I think, P less than 0.05. So to signify that, I made a different symbol for them. So these are kind of like asterisks, but since they were signifying different things, I didn't want to use the same symbol. You can use letters or asterisks or whatever floats your boat, okay? So that's something that I liked about this graph. So I'm going to ask you to do, we're going to do analyses for each of our dependent variables, which are what? Not amount of estradiol, that's our independent variable. That's what we manipulate. Dependent variable is what you measure. Area for RA, HBC, and X. So we're going to do 3 analyses, okay? And so you're going to have 3 dependent variables, you're going to have 3 analyses, you're going to have 3 graphs. All right? Make sense? Okay. All right. I will come back more and talk about results next time. Let's know when you have your data in hand. Let's move on now to discussion. Okay, so what you should do in your discussion is go back to your original purposes. So we had that list there of why we were doing this study. Okay, so you should lead off and start talking about the original purposes. So use your data then to support your conclusions, okay? So we were trying to replicate the sex differences. Did we do it successfully? Yes or no? That you can refer the reader, and you should refer the reader to a figure again, at least. You don't have to put in the analysis, but you should at least get them back to a figure so they can go back, oh yeah, sure shooting. There it is, okay? So you want to go through first and say, okay, well, here's one of our purposes. Here are the data to answer that question. And, you know, and again, 1 idea per paragraph, please. Okay? All right. So what I'd like to have you do then is go through and see, did we replicate the sex differences? And when you talk about this replication, then again, you need to be making reference to previous written things, okay? So yeah, hey, we got a difference just like Nottebohm and Arnold or Grisham and Arnold, and you can see clearly in figure 1, 2, or 3, whichever it is, maybe all of them, that we got it, or if we didn't get it, you know, that's something to talk about too. Okay, so let's look at, this is data which I am hoping your data will be similar to. All right, so did we get a sex difference here in these data? Why do you tell me these things? Okay, but I told you what the A's and B's and everything stands for, right? So what about these data explicitly show me that you did replicate the sex difference? No, B's are, all right, so once again, I must have done it too fast. A, significantly different from every other group. Okay, B, significantly different from 5 and 0, but not from each other. Okay? So, is there a sex difference? Do these data show evidence that there is a sex difference, and why? Oh, geez. A is different from the rest of the women, and most explicitly, actually, you know they're all females, but the one you'd really want to show is it's different from whom? The controls, so the zeros, all right? So that difference, pow, nails it. All right, we got it. Okay, so did we replicate the sex difference? What aspect of the data allows that conclusion? You want to walk the reader through it. Don't make us think. Don't make us think. It should be so clear we don't have to think. We hate it when that happens. We have to think. All right? So don't make us think. Does this reference the readings that this replicates? Okay? Let's see. I got a little bit of time. Let's do the next one. Gauge the sensitivity of the female zebra song system to estradiol. Okay. Is it exquisitely sensitive to estradiol? And let's see, what data allow this conclusion? And what does it say about the sensitivity of the song system in males? Okay. So here, we got these data. These, I hope, will be similar to your data. So what about the song system? Is it exquisitely sensitive to estradiol? Why or why not? Yep, and 5, as indicated in this graph, wasn't significantly different from 0, okay? So it wasn't that any teeny tiny dose would set off the masculinization, so it's not exquisitely sensitive. Okay, good. All right. By the way, you might think that 5 micrograms is a teeny tiny amount, but I don't know, did any of you ever have Dr. Schlinger? Did he talk about how steroid hormones can have impacts at microgram levels on humans? Even so, like a birth control pill has got about 50 micrograms of estradiol in it, okay, or something like it, okay? So even on an animal our size, microgram quantities of hormones can have a big impact. So you can imagine a little tiny zebrafinch that's about the 1,000th of your size. 5 micrograms is actually fairly sized dose, okay. Okay, does it say anything about the song sensitivity of the song system in males necessarily? What do you think, gang? No, doesn't have to be true in males, right? We didn't test that. Okay, one more and then I'll let you go. All right, we want to see if masculinization looks like an all or none event once the threshold is reached, or does it show a graded response, okay? So let's say you get data that look like this. What do you think? Is masculinization kind of like an all or none effect once you reach a given dose, or is there a graded response to different doses of estradiol? Okay, so let's actually talk about that. So as far as the statistics are concerned, these two groups, even though there looks like there's a little uptick, are essentially the same. All right. So any difference, the statistics say any difference there is just due to sampling error. So we can't really say that's due to our teeny tiny dose of estradiol. Can't do that. All right. So given that, does that change your interpretation? What would you then say? Yeah. Okay. And what else tells you it looks like a threshold and not graded? 50 and 50 are not significantly different. So the statistics are also saying, as far as they are concerned, these two are essentially the same. Okay? So If it was a greater response, we'd get like a little bit of a difference here and then a bigger difference for 15 and yet a bigger difference for 50, but we didn't see that. Once you hit 15, it seemed to be as good as 50. All right, and it looks like I've used my time, so I will see you in lab and talk to you more then. Thank you.